السلام عليكم. أنا الطالب هشام زاهر وأعضاء الفريق معتز كامل ليما سمارة لبنى عصام. راح نتحدث اليوم عن البايو تشيب for protein analysis. So we will talk about protein microarrays. Protein microarrays used in the field of proteomics. Proteomics is a study of all proteins that are expressed at a certain time inside cells, tissues, organs, and organelles. So we will look for how a protein profiling, how abundant a protein is, and how it's interacting with other molecules. So to do this task fast and accurately, we have certain techniques like two-dimensional gel electrophoresis, LC-MS, and microarrays. To better understand the word microarrays, we divide it into micro and arrays. Arrays are solid supports upon which a collection of gene or protein probes has been placed at defined locations. Micro refer to small size of slide. So protein microarrays are found due to some limitations in DNA microarrays, such as the concentration of mRNA are poorly correlated with actual abundances of corresponding proteins. The second one is post-translation modifications like addition a methyl group, which is critical for determining protein function, are not visible on DNA microarrays. So protein microarrays, sometimes called protein chips, is a high throughput method used to track the interactions and activities of proteins to determine their function and to determine function on a large scale. As we see in the figure here, at each spot we know what is present and if there is a detection or not. So which means that a particular protein is present in your sample or not. The big advantage of using protein microarrays is a huge number of proteins can be tracked in parallel. Here we will talk about the principle of protein microarrays as we see here. So you take a support system, which is a slide, as we see in the step number 1 here. Then allow the protein to immobilize on that slide, and then we design a probe. Very important thing is labeling. Labeling either by fluorescent labeling or radioisotope labeling. After labeling, antigen and antibody reaction take place between the complementary molecules, as see in the step number 4 here. So if we use fluorescent labeling, after binding, there will be fluorescence generated. So the one which have been binded will flourish. So which means that they will emit some kind of signals in the form of color, as we see here in the step number 5. So then after detection, the data is generated, as seen in the step number 6, and this can be interpreted by the expertise in the field.
I am Mu'taz Mekawi. I will talk about the types of protein microarray. There are 3 main types of protein microarray that are used to study the biochemical activity of proteins. First, analytical microarray, and second, functional microarray, and third is reverse phase Analytical protein microarray used to profile a complex mixture of a protein in order to measure binding affinity, specificity, and protein expression levels of the protein in the mixture. What we do is we prepare an array library of antibodies optimers, which is a short and a single-stranded DNA or RNA molecule, or affibodies, which are a small molecule that have a high affinity toward a specific protein target. This useful molecule have the application in therapeutics, diagnostics, and biotechnology into a glass microscopic slide, and then this array is probed with a protein solution. Analytical protein microarray used to monitor different expression profile and for identification of biomarker for versus disease and for clinical diagnostics purpose. Examine to profiling response to environmental stress and healthy versus disease tissue. One of the most common analytical microarray is antibody microarray. The functional protein microarray, which is differ from the analytical arrays. The functional protein arrays are composed of arrays contain full-length functional protein or protein domain. This protein chip are used to study the biochemical activity on an interproteome in a single experiment used to study of numerous protein interaction inside our body, such as protein-protein interaction, protein-DNA interaction, protein-RNA interaction, protein phospholipid interaction, and protein small molecule interaction. The third type of protein microarray related to an analytical microarray is known as a reverse phase protein microarray. Here the cells are isolated from the versus tissue of interest and are lysed. The lysate is arrayed into a nitrocellulose slide using a contact pin microarray. The slides are then probed with antibody against the target protein of interest, and the antibodies are typically detected with chemoluminescent fluorescence or colorimetric assay. Reverse phase protein microarray allow for the detection of the presence of altering protein that may be a result of disease, especially post-translation modification, which are typically altered as a result of disease, can be detected using this technique. Once it is determined which protein pathway may be dysfunctional in the cells, a specific therapy can be determined to target the dysfunctional protein pathway and treat the disease of interest.
Regarding the applications of the protein microarrays, we found that the two major parts that were involved in the applications are diagnostics and proteomics. Now, first of all, regarding the diagnosis, this involves us detecting the antigen and antibodies in a blood sample. Moreover, we can use them to discover new disease biomarkers. Now, a biomarker is basically a way for us to see how well our body responds to a certain treatment of a certain disease. And thirdly, we can use protein microarrays to discover or to monitor the state of the disease which we are in. And if you were interested in personalized therapy, it can be used to see how effective it is. Secondly, we have the characterization of antibodies. This includes the epitopes mapping, cross-reactivity, as well as the specificity of a particular pro-antibody. Thirdly, we have the proteomics. طبعا هون بنقصد فيها البروتين expression profiling بمعنى how abundant a certain protein is inside the cell. Functionality wise as well, how exactly does it interact, what kind of interactions does it have, or what kind of interactions take place. The fourth application that we're going to discuss is the protein functional analysis. What do we mean by the protein functional analysis? It is basically a tool that is used to assign biological or biochemical roles to proteins. طبعا اللي بصير إنه we immobilize the purified proteins. In that way we can identify the protein-protein interactions, the protein DNA with the protein RNA, and so on, as well as the enzymatic substrates and the receptor ligands. Last but not least, the protein microarrays have proved themselves in treatment development. Now this is kind of a subsequential result, since we already talked about the fact that we can use protein microarrays to identify biomarkers. Now we know the way our bodies respond to a certain treatment, hence we can work on developing this treatment. Moreover, it has been used in assessing transcriptional activities in cells that were subjected to therapy. فبالتالي we now have something to look back on. In addition to development of antigen-specific therapies for autoimmunity cancer, for allergies, and so on.
Challenges. In this part, there are certain challenges. As well, firstly, they don't have longer shelf lives because proteins are complex and maintaining their native state. In their secondary and tertiary structure, it's a little difficult because they require wet environment, aqueous environment. So to maintain their proper structure, that is of a huge importance. The second one is improper binding. Sometimes improper binding takes place when you are fluorescently labeling. These fluorescent dyes sometimes bind to the active regions of the protein and it will hinder the binding the target protein with the active region of that protein. The last one is antibodies. As we know, antibodies are commonly used as probes in protein arrays. For antibody microarrays, the antibodies must remain active to bind to the analyte on the surface. This requires an immobilizing material that maintains the active site of the antibody for long periods of time in storage. There are two big challenges. The first one is loss of activity. That is because the active binding site may bind to slight surface through chemical bonding, and thus the active site may not be available to the antigen. And the second one is cross-reactivity. Specificity is also a big issue for antibody protein arrays that occurs when the proteins in one substance are like the proteins found in another substance.